People that detect other tests only analyze a few specimens at a time, maybe less than 20, but are a little faster. It really depends on the different tests. Here at Mayo Clinic, we use two different tests for sequencing. One is done in our clinical laboratory, and that is the Genexus test, the Genexus instrument by Thermo Fisher. We use that for testing clinical patient specimens, and we use that mostly for hospital infection and control, and also just general information about the variant the patient has. It doesn't impact treatment at this point. The other test that we use is done in the research laboratory by Dr. Wieben and his lab, and that has a much larger capacity. We could sequence 700 specimens at once.
Now, many of the people watching this talk by now will have had 1, 2, 3, or even 4 doses of a messenger RNA vaccine. With billions of these shots now in arms, it's clear that this new way of making vaccines is both remarkably safe and incredibly effective. But did you know it's not the vaccine itself that's keeping you safe? It's actually you. Because the human body has amazing powers to both prevent and cure disease by making its own medicines. You just need to know what medicine to make. And that's what your vaccine gave you, simply a set of instructions for how to protect yourself against SARS-CoV-2. Now, vaccines are only the beginning. As said before, the advent of mRNA vaccines is heralding in an entirely new era of medicine. mRNAs give us the ability to not only prevent disease, but also treat previously intractable disorders. But before I get to that, let's talk about really what is this new way of medicine really about? Well, it all comes down to protein. Now, you may think of protein as simply something that you need to eat, your diet, something that's important for you to build muscle. But it's not just muscle that contains protein. Protein is a huge fraction of the incredibly complicated ecosystem that's your entire body. In many ways, your body functions like a large city, full of myriad buildings, interconnected buildings, with lots of different structures. Now, just as the word building fails to capture the incredible variety of structures that make up any large city, the generic term protein gives no clue as to the incredible variety of molecular architectures at the molecular level. Like buildings, proteins are not monolithic. Our body makes many proteins. Some like collagen in our skin that makes our skin tough but pliable. Like the actin and myosin in our muscles that enable us to move. Or our blood is full of hemoglobin, oxygen around. Antibodies protecting us from disease. And clotting factors that close up our wounds. And for any of you that have ever cracked a biochemistry textbook, this should look familiar. This is the metabolic chart of the human body. These are all of the reactions in your body that are keeping you alive and well right now. And each one of those reactions is catalyzed by a different protein whose names are shown here in blue. So proteins are not only what make your body, what make up the bulk of your body, they're also what make your body tick and keep you well. Now, if we think again about buildings, the structures of buildings may look quite different in the end, but they're all made of a limited set of building materials. And how those building materials, which of those building materials are used, and how they're arranged, and how they're attached to one another, gives the buildings their final form. The same is true of proteins. If we zoom down to the molecular level, we can see that proteins,
Hello and welcome to Strand Explains, a podcast where we discuss what's happening in the world of precision medicine. From weird case studies and interesting facts to interviews with leaders in the industry.
I'm Sakshi, and today I'm joined by Dr. Ramesh Kumar.
V2. Then you're subjected to something called extraction, where you extract out the virus's genetic code and purify it, along with all of the human DNA and RNA as well. And the PCR, in the case of the tests we're using at Mayo Clinic, are tests that amplify little pieces of the
messenger RNA. Your body takes the messenger RNA in the food that you consumed, breaks it down into those components.
ಮತ್ತು ಇನ್ ಕೋವಿಡ್ ಸಪೋರ್ಟ್ ಸಂಸ್ಥೆಗಳಿಗೆ ವಿಶೇಷ ಧನ್ಯವಾದಗಳು. ಕರ್ನಾಟಕದಲ್ಲಿ ಕೋವಿಡ್ ಜಿನೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸಿಂಗ್ ಮತ್ತು ಸರ್ವೆಲೆನ್ಸ್ ಕೆಲಸದಲ್ಲಿ ಭಾಗವಹಿಸಿದ ನಿಮ್ಮೆಲ್ಲರನ್ನೂ ಸ್ವಾಗತಿಸುತ್ತೇನೆ. ನನ್ನ ಹೆಸರು ಪ್ರದೀಪ್ ಮತ್ತು ನನ್ನೊಂದಿಗೆ ಮೌಶಮಿ ಈ ಸಂಜೆ ನಿಮ್ಮ ನಿರೂಪಕರಾಗಿರುತ್ತೇವೆ.
Good evening, ladies and gentlemen. A warm welcome to all of you. While we are waiting for our special guest to come, we are starting with our first speaker, Dr. Rakesh Mishra, currently the director of Tata Institute of Genetics and Society. Dr. Rakesh received his master's and PhD degrees from the University of Allahabad and did postdoctoral research at the Indian Institute of Science, the University of Bordeaux, St. Louis University, and the University of Geneva. He joined the Center for Cellular and Molecular Biology, or CCMB, in 2001 and became its director in 2016. His research interests are in the comparative genomics of non-coding DNA. Dr. Rakesh is a J.C. Bose Fellow and a Fellow of the Indian National Academy of Sciences, the Indian Academy of Sciences, the National Academy of Sciences, and the AP Academy of Sciences. Dr. Rakesh has been a key member of the Indian SARS-CoV-2 Genome Sequencing Consortium, or INSACOG, and has closely tracked the evolution of the virus. I request Dr. Rakesh to share with us his experience as part of the INSACOG effort. Since Dr. Rakesh will join us virtually, I request our technical team to please connect us to him.
I'm going to focus mainly on the genomic surveillance, which is essentially constantly monitoring pathogen and analyzing the genetic similarities and differences, which is very important. And why we should do that? We should do that because we can then track the spread of the variants, monitor the changes to genetic code of the virus, and collectively this information is then used to better understand how variants might impact public health, which can help us design strategies to address the problem. And it's also helpful for researchers, epidemiologists, and public health officials to monitor the evolution of the infectious diseases and alter the spread of the alert the on the spread of the pathogen and develop countermeasures like vaccines. We need to do that particularly in this case in the SARS-CoV-2 because it mutates very frequently. Mutation happens when virus is replicating inside the host, which is human, which means more and more people infected, more and more mutations are likely to come. And mutation can happen actually do happen by chance, but they are selected based on fitness. If it is advantageous to virus, represented the variant will be represented more. If it is disadvantageous to virus, which is often the case, it will be wiped out from the population. And just to make a point that more mutation does not necessarily mean virus more dangerous. This virus has been accumulating mutation roughly at the rate of two mutation per month. And this is the map of the virus. Here is the spike protein. And as you see that there are more number of mutations recorded there. That is because virus uses this as a key to open the lock to enter the host cells. And the host cells tries to defend itself by destroying the key by, let's say, by the antibody. So lots of catch-22 goes on at this point of genome of the virus. So in this context, what should be the strategy for the genomic surveillance? Sequencing as many samples as possible sounds very reasonable thing, but number of sequences, where do we stop? What percent we should, how much is enough? And factors that influence the outcome is not necessarily numbers, as I will show you some of the observations. What we have to keep in mind is that what is the selection pressure on the virus, why it is evolving, and what is being putting the pressure for the selection. So one is higher infectivity. So if more infectious, it will have advantage. Second is immune escape or vaccine breakthrough. In current scenario, almost everybody has got either infection or vaccine and boosters and so on. So virus is desperately looking to break those protections. Mortality and worse symptoms are not any advantage to the virus. If new variants come with that, that's simply our bad luck, not advantage to the virus. So the strategy for genomic surveillance should include when we decide to collect the samples in long term as a country, when we are planning this, what we have to sequence is the unusual clinical symptoms or hospitalized cases, because this may be that either the person has comorbidity or some unique problem, or a new variant is appearing. So that's very important to collect those samples for sequencing. Vaccine breakthrough cases should be sequenced. So it could be that the vaccine is fading or not working, or a new variant has emerged. Reinfections are important to sequence. So that will tell, is it the immune deficiency in the individual, or fading immunity, or a new variant has emerged which does not care about the vaccination, earlier vaccination. Local super spread event incidents when happen, they should be sequenced because it will rule out whether it was a human behavior, people at parties or marriage parties or large gatherings, or a new variant has come which is making super spread events. And in addition to all these, being a large country, there has to be a geographically randomized sampling so that we are able to keep track of the trend and the new variant that might emerge. We should know that variants are not going to emerge only in the big cities. So we have to somehow cover geographically randomized samples from all over the country, which is a huge problem for our country, being such large in size. Just to give you a glimpse of that, we have been doing this sequencing for quite some time now and made a dashboard where you can go and see year 19, which tells you how many sequences from India have been deposited in the public domain, so close to 2 lakhs. 51,000 mutations have been catalogued, and so many lineages are being traced, and 36 state data is included in this. And then you can play with what lineage is doing well, and so on. And this also tells us over a period of time how the lineage scenario is changing. Each color means different lineage. And as you can see, some is emerging, then fading away, then a new variant is emerging, fading away, and so on. And this is now the pink one is BA.2, which is mostly the case, the variant in the country. And as you can also see that at one time it is Delta, which is showing its presence here. Now it is a BA.2, which is showing its presence here. One thing we have to keep in mind, which is a big problem with us, is that this does not mean that the states which are darker have more cases. Other places, sequencing is not that much, and that's why what we are plotting is not really reflecting the reality on ground. So that is one thing that needs to be fixed. Having said that, now how we are doing at in the global context. So this color on the top is the USA, the America. You can see that most of sequencing is done there, and then the Europe, and then that little dark line that you see is the Southeast Asian region. So little contribution of sequencing that we are doing at the Southeast Asian region. Similarly, cumulative proportion of cases sequenced by the country, if you see, India is very, very low, one of the lowest actually. However, if we look at the earliest identification of the various variants like alpha, beta, gamma, delta, and omicron in each country, now you see that we are almost at the same time as any other country who has been sequencing like crazy. And it is consistent all the way up to here. You see, we are far ahead of many other things. So which means that the number of sequencing is not necessarily the only criteria. How you are sequencing and what is your strategy, that also decides. And looks like from this observation, which came recently in Nature Genetics, that we have done well, picked things as early as others have done. So that was about the patient sample sequencing. I will take next 5 minutes to explain you a little bit about the environmental surveillance, which is emerging out to be, I would say, one of the booms from this pandemic for the healthcare system. So you can do environmental surveillance by 2 means. One is the wastewater-based surveillance and one is the air surveillance. I'm not going to talk today about air surveillance. I'll talk only about the wastewater surveillance, which is unbiased and gives early warning. I will show you some of the data for that. It's very convenient and cheap because you just have to go and collect water from STP or open drainage. You don't have to contact any person. And instead of testing thousands and thousands and thousands, you just have to collect a sample from the water source, and then very few of them are there, which will be enough. And this process is relevant even beyond SARS-CoV-2. You can actually monitor various other pathogens, including antimicrobial resistance responsible genes. And I'm going to next few minutes talk about data which has recently been deposited in medRxiv, and I'll urge those few who want to learn more technical and details of this can go and take a look at this. So we are testing all the 28 STPs of the Bangalore city, and this is not possible without the collaboration, cooperation of large number of organizations, including BBMP and BWSSB, which is extremely important, and of course the funders. And what I'm going to talk about is the work recently done by Paraj Tiag, Sanjay Lamba, Kumar Ramakrishnan, and Mr. Vishwanath, who has been extremely helpful and encouraging for this study. So here is one observation to show you that wastewater surveillance reveals the secrets of the SARS-CoV-2. The red one is the COVID-19 positive cases in the entire city, and the blue is the cumulative normalized viral load from the STPs. You can see this is the third wave. You can see that you get little early warning of the third wave. And these are not false peaks. These peaks were never noticed. This is actually Dasara peak during that time. This is now the current increased number of cases. So this is what you see, and this is what actually estimated by wastewater. So we know how cases are increasing or decreasing well before actually it shows up in the population in terms of symptom and testing. Here is more accurately shown. If you take a lag time of 7 days, you can have actually 14 days advance notice. You can see the blue line coming early. And if you take time lag of 4 days, you can get up to 8 days of advance information that what is going to come. And we have now tested in many different ways, and it's really reliable. What is more interesting is that it's not just the whole city. Here, you don't have to worry too much about what is written in it. Just shows that each set of these pictures is each STP. And we know for each STP how the cases are going up and down, and these are the variant scenario there and along the different timeline. So this shows that maybe in some places you will see more cases here, while in this STP there's less case. We know what are the catchment area, what are the ward associated with this. So a special action may be needed only in those places and not in the entire city like Bangalore, which is a big city. So this is extraordinarily granular and scientifically robust data that helps us take strategic decision for controlling the situation before it gets too worse. So this is an advantage of this method. And here we are comparing, so we take the samples, not only estimate the number, but we also do the sequencing and get the variant scenario. As you can see, this is from beginning this year up to the latest data. And this is the data from the individual sample collection and sequence and plotted along the same timeline. So you can see by and large, you will see similar variants coming. But you see in wastewater, you see generally more colorful graph, which means there are more variants that we detect in the sewage system because we are sequencing, sampling the entire population, while here we are sequencing only who we have collected the samples. So that's severe limitation. So while this is extremely important, of course, because the quality of data is different than here, but this gives you really a global picture of at STP level of granularity. So one thing which I forgot to tell here is that as you see here, one color, this shows that there are a lot of unassigned entries, which means we don't know which variant it is. Yes? Yes, you want to talk, Harshit? Hello? Yes. Yeah, tell me. Okay, I think she wanted to say something. So what we are seeing is a lot of unassigned variants, which is something very peculiar thing. And we wanted to know what are these unassigned entries? Are these sequencing errors? Are these bioinformatic limitations? Or these are unmappable SARS-CoV-2, which probably is a recombinant. So if it is a recombinant, which some of you may have heard in the newspapers, is it a new trick of the virus? If so, then how successful it has been, and does it mean more deadly prospects we have? So we have focused on this part, and we show that.
I'm sorry to interrupt, Dr. Rakesh Mishra. Actually, I would request you to please complete your talk in the next 3 minutes, if that is possible.
Yes, I'll finish.
Thank you so much.
Yeah. No problem. Yeah. So you can see that a large number of, and the number of unassigned variants actually peak during when the cases are more. So what does it? So but we really did this analysis to show that this is all computation analysis, and we see if these consider them as a potential recombinants, then we can see that this recombinant will be generated. One sample is from Jammu and Kashmir, one is from Delhi, and their gap in collection date is 1 month, and these are the variants. So now you see that they are from the same place and at the same time. So that means two different variants are same place and same time, which have infected one individual and made recombinant. So it's a very powerful way of showing that recombinants are happening in this virus. So is it new trick the virus? Yes. So far, has it been successful? No, because these recombinants don't make too much of a headway, and they are detected, but they are lost. But we cannot write it off straight away, because this is a new trick that the virus has. And this is essentially shows that from the CVS surveillance, you can detect the variant composition and the dynamics as we've been seeing with others. And this is we are towards the end. If you see that in past few months, past 6 months, in fact, from the CVS surveillance, you can tell what are the variants that are like here is the BA2, BA2.10. And interesting thing is there are recombinants also which are hanging around, and hopefully they'll not become a problem. So what is the take home from this is that wastewater surveillance is an effective method for genomic surveillance that is essential. It also complements the patient sample surveillance. So I would like to then thank all of you, thank CCMB for the early support for this study. And my special thanks are to BBMP and BWSSB. It was not possible, it is never possible unless we have full cooperation and support of these two organizations. And by this together, we are setting really an example for others to follow. So we look forward to working together and making our study as a model, which can then be adopted to other cities. Thank you very much. And if there are any question, clarification, happy to do that. Thank you very much.
Thank you, Rakesh Mishra. Thank you, Dr. Rakesh Mishra, for such an insightful talk. Now, we would like to call our next speaker. Our next speaker is Shri Sandeep Nelwal, co-founder of Polygon, the world's biggest layer 2 protocol operator for the Ethereum blockchain. Sandeep graduated with an engineering degree in computer science and worked as a software technologist before he founded Polygon. Polygon was founded to offer a protocol that would make Ethereum transaction cheaper and faster. It has about 7,000 decentralized apps, also called DApps, with more than 130 million unique users and 3 million daily transactions. Seeing some of his relatives seriously affected by the second wave of COVID-19 in India, Sandeep called upon his network and set up the COVID support organization. To date, the fund has raised over $440 million, which it has distributed to NGOs and other agencies working on the ground for healthcare relief in India. I request Shri Sandeep Nelwal to tell us more about the fund.
Hello, everyone. First of all, thank you so much for having me here. About Crypto Relief, as the host already introduced the origins of the fund, but as an organization, Crypto Relief is committed to strengthen the genome sequencing activities in India. Apart from a lot of other things that we are doing, we feel that the genome sequencing and early detection of these outbreaks is the most important thing. We were fortunate enough to support Strand Life Sciences in carrying out genome sequencing in this scale, and we look forward to contribute more in this regard. Crypto Relief has also supported infrastructure related to genome sequencing in IISc, Bangalore, IIT Madras, LNJP Hospital, Delhi, and other such places. Looking towards the future, we plan to establish Indian Alliance for Environmental Surveillance, IAES, in which we are looking to partner with various government and private institutes of prominence in India. And one of the major activities of this alliance will be genome sequencing of various pathogens likely to cause health concerns in India. We are also investing in projects that will help build an open bioinformatics pipeline for Indian facilities, whose initial focus will be COVID and antimicrobial resistance. But overall, eventually we want to expand it into overall virology activities, virology detection related activities and other things. We also envisage a state-of-art virology institute in near future in India, which will serve as a core facility for all virology related activities carried out by Crypto Relief. Once again, we congratulate Strand Life Sciences for their excellent work and wish them more success in future. Thank you so much.
Thank you, Shri Sandeep Nailwal.
ಮಹಿಳೆಯರೇ ಮತ್ತು ಮಹಾನೀಯರೇ, ಈ ದಿನದ ಶುಭ ಸಂಜೆ ತಿಳಿಸುತ್ತಾ ಇಂದಿನ ಕಾರ್ಯಕ್ರಮದ ಉಪಸ್ಥಿತರಿರುವ ಕರ್ನಾಟಕ ಸರ್ಕಾರದ ಗೌರವಾನ್ವಿತ ಸಚಿವರುಗಳಾದ ಡಾಕ್ಟರ್ CN ಅಶ್ವಥ್ ನಾರಾಯಣ್ ರವರಿಗೆ ಮತ್ತು ಆಗಮಿಸಲಿರುವ ಕೆ ಸುಧಾಕರ್ ರವರಿಗೆ ಹಾಗೂ ಬೃಹತ್ ಬೆಂಗಳೂರು ಮಹಾನಗರ ಪಾಲಿಕೆ ವತಿಯಿಂದ ಆಗಮಿಸಿರುವ ವಿಶೇಷ ಅತಿಥಿಗಳಿಗೆ ಮತ್ತು ಎಲ್ಲ ಗಣ್ಯರಿಗೆ ಹೃದಯಪೂರ್ವಕ ಸ್ವಾಗತ. ಈ ಕಾರ್ಯಕ್ರಮದ ಪ್ರಮುಖ ಪಾಲುದಾರರಾದ ಮತ್ತು ಸಾಂಕ್ರಾಮಿಕ ರೋಗವನ್ನು ತಡೆಯುವಲ್ಲಿ ನೀಡಿದ ಬೆಂಬಲಕ್ಕೆ ಬೃಹತ್ ಬೆಂಗಳೂರು ಮಹಾನಗರ ಪಾಲಿಕೆ, ಸ್ಟ್ರ್ಯಾಂಡ್ ಲೈಫ್ ಸೈನ್ಸಸ್ ಮತ್ತು ಇನ್ ಕೋವಿಡ್ ಸಪೋರ್ಟ್ ಸಂಸ್ಥೆಗಳಿಗೆ ವಿಶೇಷ ಧನ್ಯವಾದಗಳು. ಕರ್ನಾಟಕದಲ್ಲಿ ಕೋವಿಡ್ ಜಿನೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸಿಂಗ್ ಮತ್ತು ಸರ್ವೆಲೆನ್ಸ್ ಕೆಲಸದಲ್ಲಿ ಭಾಗವಹಿಸಿದ ನಿಮ್ಮೆಲ್ಲರನ್ನೂ ಸ್ವಾಗತಿಸುತ್ತೇನೆ. ನನ್ನ ಹೆಸರು ಪ್ರದೀಪ್ ಮತ್ತು ನನ್ನೊಂದಿಗೆ ಮೌಸುಮಿ ಈ ಸಂಜೆಗೆ ನಿಮ್ಮ ನಿರೂಪಕರಾಗಿರುತ್ತೇವೆ. ಥ್ಯಾಂಕ್ಸ್.
Thank you, Pradeep. Good evening, ladies and gentlemen. In June 2021, Strand Life Sciences, Karnataka State and BBMP and philanthropic organization in COVID support and other organization like ACT COVID Response Collective came together to step up genomic surveillance efforts. The main objective of this collaboration was to facilitate early detection and assessment of both local as well as imported variants of concern and provide timely analysis of outbreaks that can inform public health policy. Strand Life Sciences has since performed genomic sequencing of over 12,800 COVID samples in Bangalore and Karnataka over the last year, supplementing INSACOG's sequencing effort. The result of these sequencing efforts were translated into actions on the ground via close collaboration with the Karnataka State and the BBMP public health officials. Over the next 2 years, all the stakeholders in this exercise will share their experience and learning so that such collaboration can grow and make our pandemic preparedness stronger. First, I would like to introduce our speakers for the evening. Dr. Ashwath Narayan, Honorable Minister of Electronics, IT/BT, Science and Technology, Higher Education, Skill Development, Entrepreneurship and Livelihood, Government of Karnataka. Dr. K. Sudhakar, Honorable Minister of Medical Education, Health and Family Welfare, Government of Karnataka. Dr. K.V. Trilok Chandra, IAS Officer of Karnataka Cadre and the Special Commissioner, Health and Animal Husbandry, BBMP. Dr. Balasundar, Chief Health Officer, BBMP. Dr. Rajesh Sundaresan, Professor in the Electronics and Communication Engineering Department and the Dean of the Division of Electrical Engineering, Electronic Engineering at Computer Science at the Indian Institute of Science. Dr. Shri Naga Karthik in COVID support. Dr. Vishal U.S. Rao, Director, Head and Neck Oncology, SCG Cancer Hospital, Member of Genomic Surveillance Committee, Government of Karnataka. I welcome all the speakers, a warm welcome to this event. Now I would like to request Dr. Ramesh Hariharan, co-founder and CEO of Strand Life Sciences, to please felicitate all our honorable special guests. Dr. Ashwath Narayan, Honorable Minister of Electronics, IT, BT, Science and Technology, Higher Education, Skill Development, Entrepreneurship and Livelihood, Government of Karnataka. Please give a big hand for Dr. Ashwath Narayan. Dr. Vishal U.S. Rao, Director, Head and Neck Oncology, SCG Cancer Hospital, Member of Genomic Surveillance Committee, Government of Karnataka. Please give a big hand for Dr. Vishal Rao. Dr. Bala Sundar, Chief Health Officer, BBMP. Please give a big hand for Dr. Bala Sundar. Dr. Rajesh Sundaresan, Professor in the Electronics and Communication Engineering Department and the Dean of the Division of Electrical Engineering, Electronics Engineering and Computer Science at the Indian Institute of Science. Please give a big hand for Dr. Rajesh Sundaresan. Shri Naga Karthik in COVID support. Please give a big hand for Shri Naga Karthik. Thank you, Dr. Ramesh, and welcome again to all our speakers. I would like to request Dr. Ashwath Narayan and Dr. Vishal Rao to please come and do the honor by lighting the lamp. Thanks to all our special guests. Before we move ahead with the talks, I request the audience to stand up and observe a 30-second silence for the lives lost to COVID and for the frontline efforts from various constituencies to battle this pandemic. Thank you, everyone. Please take your seats and be comfortable. We will now begin with a welcome note and overview of Strand genomic surveillance efforts. We will then request the Honorable Minister, Dr. Ashwath Narayan, to give his special address. We will then hear from Dr. Vishal Rao, followed by Shri Naga Karthik, and Dr. Vamsi will conclude with a vote of thanks. So without further delay, I would like to introduce our first speaker, Dr. Ramesh Hariharan, co-founder and CEO of Strand Life Sciences. Please give a big hand for Dr. Ramesh Hariharan.
Welcome to everybody and welcome to all the guests out here. I'll take a few minutes to just set the stage, and then we'll hear from all of our invitees. This project that we've conducted over the last year is a very unique project in the sense that the number of stakeholders that had to come together is, I would say, relatively unprecedented. The genesis goes back to a year ago when we all faced the wrath of the Delta virus. And that happened late March, early April is when we really saw the virus, shooting up. However, while genome sequencing efforts were in relative infancy at that point of time, there was sequencing happening then and prior. And what was clear is that even from the data from India that exists, that in the month of February, going into the month of March, the virus went up from 0.1%, the Delta strain went up from about 0.1% to cross the 1% mark. And that was about a month before it really, the effects started becoming apparent. And that got us all thinking that that's what we need to aim for. We need to catch strains when they're at about 0.1%, going towards the 1% mark. And that will give us at least a month's leeway, if not more, before we can act. And if you look at it even going farther back in time, all the way back, genome sequencing data from India and Asia shows that all the way back in October, The virus, traces of the Delta virus were there. However, they were at levels which are hard to act upon. Even today when we sequence, and you'll see this, we do find many new strains there at very relatively low numbers. And you don't necessarily need to act upon those because they will go away on their own. This one, of course, did not. And it started crossing the 0.1 to 1% mark sometime in the February timeframe. And that's what we need to watch out for. And this got us thinking as to how do we make sure that we have enough surveillance in place to do this sort of tracking. INSACOG was there. It was relatively at a much lower scale then. And several parties got together to say, how do we supplement and make this much larger? And so that's where this partnership came about. And as part of this partnership, I'll tell you who the partners were, but as part of this partnership, the goal was to focus on Bangalore and Karnataka, largely Bangalore and a little bit more beyond Bangalore to Karnataka, pick up RT-PCR positive samples from various labs in the city, and some of them from the rest of Karnataka, and sequence them on a regular basis. And as we saw, we have to track from 0.1% to 1%, which means we have to be sequencing roughly about 1,000 cases every month to get hold of the 0.1%. And so what we did was these samples were collected and brought into labs. So InStem, which is an INSACOG lab, was functional. Strand added on to that capacity. And there were other labs that came about eventually. And then all of that sequencing, the results of that on a weekly basis or a bi-weekly basis were then communicated to BBMP and to the government of Karnataka. And these were the sort of agencies driving action on the ground with the information that we would provide. And all of this needed to be funded. And that's where you heard Sandeep, who's the founder of the In-COVID Support Fund. In-COVID Support stepped in with enough funding to supplement the INSACOG efforts. INSACOG efforts were funded by the government, and then this was privately funded. And there were others as well, in addition to In-COVID Support, who was the main funder. There were other smaller sources of funding. The ACT COVID Response Collective also contributed. And all of this came together between the sequencers, Strand and InStem, BBMP and the government of Karnataka, as well as the philanthropic funders. This is a unique instance where all three parties came together, or a relatively unique instance, where all three parties came together to supplement government efforts and provide intelligence for action on the ground. There were, of course, other efforts that were ongoing. BBMP and the government of Karnataka was doing sero surveys, and various COVID modeling efforts were ongoing. And that's where Indian Institute of Science and ISI came in. So we have today people from all of these. I'm talking about what we learned from Strand. Government of Karnataka, Dr. Ashwath Narayan is there. Hopefully, we'll hear from Dr. Trilok Chandra as well from BBMP shortly. We heard from Sandeep Nelwal on the In-COVID support. And we'll hear from, and of course, Naga is there to talk more about In-COVID support. And we will also hear from further on BBMP. And we've got Rajesh Sundaresan from the Indian Institute of Science to add perspectives on the sero surveys and the COVID modeling work that happened. So all of these organizations sort of came together to actually do a lot of multidisciplinary activity towards managing COVID. I'm going to just walk you through what we learned over the last 1 year, in month by month on our sequencing and how that worked. So we aimed to do about 1,000 samples every month, which was the 0.1% number, tracking number that we aimed for. Of course, some months we fell short, various logistical and considerations, etc. But several months we were able to hit 1,000, and in January, which was a peak month for reasons we know, we actually sequenced quite a lot so that we could see what was really going on. Overall, 12,800 samples got sequenced, which supplemented the INSACOG efforts. So if you look at GISAID, which is a public database of all the sequences deposited, about 30,000 odd sequences are there from Karnataka, of which 12,800 or about 11,000 from the 12,800 have been submitted by Strand. So about 35% roughly that we submitted. And I'll just walk you through month by month, what we learned. So we were able to start this, unfortunately, only after the Delta wave had really ended or was subsiding. July was the first month when we started this, July of 2021. And not surprisingly, what you find in July of 2021 is that the Delta strains dominate. B1.617.2 was the classic Delta strain that led to the mayhem. But along with that were various other substrains. They all go under the name of AY something or the other. And together they occupied 60-30, 65-35, and they occupied the 100% pie in July. And then that picture more or less continued in our sequencing monthly, August, September, October. There were substrains that were changing. Some were going up, some were going down. But at a top level, the Delta B1617.2 and AY strains were more or less status quo, jostling amongst each other and holding status quo. There were a few unassigned strains. As we see, these 0.1% strains always show up. There will be a few of these. You don't know what to do with them. Are they going to shoot up, or are they just dying out? And we keep our eye on them. And of course, none of these really panned out later. There were, in October, for instance, there was this concern that the AY 4.2 variant in England was creating a big furor, that it's probably the next variant of concern. It might be even more powerful than Delta. Our surveillance luckily showed that there was very little AY 4.2, and so there wasn't that much concern on that count. And similar such stories. Right? Now, November was an interesting month, where if you look at the picture, nothing much changes. The Delta strains are more or less holding. There are more unassigned new strains or unknown strains, etc., showing up, perhaps errors, perhaps something that we need to wait and watch. But most importantly, the first case of Omicron shows up. It was just 1 or 2 cases in November, and also travelers coming in from abroad, mostly South Africa. So it's barely visible on the screen. It's just a sliver in blue. It's barely visible. You can't see the color. But that first case of Omicron was caught in our sequencing, and we knew that Omicron had arrived. South Africa had seen it for a month prior. And then the question was, what would happen? Delta is pretty strong. Omicron is just the first few cases are coming up. And all through December, we were on our guard. The Delta strains were still largely holding, but you could see that there is a decline starting. And that some of these other unassigned strains, new strains, etc., are not really growing. But you can see the growth happening on the Omicron strains. The BA1 strain grows rapidly from, it was barely a sliver in November, it grows to 3.6% in December. The Delta strains are holding up, but BA1 is starting to rear up. And BA2, which is the other Omicron strain, also at a lower percentage, but visible on this. So both of those from nowhere, from barely a couple of cases in November, have grown. So clearly they're starting to mount the challenge. And that was December. And then comes January, and this was a truly dramatic month, and we sequenced about 6,000 samples in this month trying to see what is going on. And you can see the dramatic change that happens. Delta, which had held forth for several months prior, was the dominant strain. In November, the first strain of BA1, December, BA1, BA2 starting to mount the challenge. In January, they just steamrolled Delta out. BA2 explodes, BA1 holds its own as well, to a lesser extent, the blue. But BA2, the BA2 strain, has really taken up most of the bulk. All the Delta strains are down to 3% from greater than 90% in just the course of 1 month. Somewhere along the way in November, December, as schools were opening, there were other anecdotes where outbreaks were happening in different places. And the question was, are these outbreaks a result of, for instance, Omicron starting to flare up? And by sequencing, we could figure out that these are actually local outbreaks, various smaller Delta strains, substrains. They're not related to each other, and therefore not yet a cause of a larger scale concern. There was a new strain as well that showed up in January, the BA3, that's the third Omicron strain that showed up in a small amount. But it's clear that the BA1, BA2, and BA3 strains are pretty much taken over from Delta. Delta is on its way out. And what is dramatic about this, that Delta took, to go from 2% to 10%, it took a month, right? While Omicron went from 2% to 86% in a month. That's how much more transmissible it was. And then February, What do we have in February? So Delta is more or less gone. The BA2 strain is dominant. BA1 and BA3 are still there. BA1 has actually started showing a decline. And for the next 3 months, March, April, May, BA2 emerges as the dominant strain. It's actually 13. The statistics are that among household contacts, BA2 has a 13% transmissible rate, while BA1 has a 10%, and that extra 3% really ensures that BA2 stays on top, and BA1 is, either minimal or on its way down. BA3 also a small amount. And then March to May, there are other smaller strains. Again, none of them seems to be expanding as of June. In somewhere along the way in April, BA5 has made an appearance in green there. It didn't show up in May, perhaps because we didn't sequence enough. It's probably there in the background. And then in the last month, June, so we're talking about data from July 21 to June 22. So in the last month of June, we've seen now the next challenger coming up. BA2, which had unseated Delta almost largely by itself, has now started to decline, and BA5 has started its growth path. And July data we don't have yet, and so what do we expect in July? There is also BA4 in June, which is a cousin of BA5, and together BA4 and BA5 are starting to mount the challenge. What do we expect in July? Well, from the US in June, we know that BA5 went from less than 10% at the beginning of June to more than 35% over the course of June, and BA2 is reducing. So we would expect at this point in July that BA5 takes over from BA2, which is, we don't have that data yet. We need to, probably INSACOG labs might have early data on that, but that's what we would expect from what's happening in the rest of the world. What's also happening in Europe is you can see that on account of BA5 and BA4, there is a slight upward trajectory that's happening. So something that we need to watch out for, which means that we need to continue to be on our toes and continue to, mount the surveillance that we need to. What is also remarkable is what is called immune escape, which is how well are vaccines and prior infection able to control these strains. As you can see from the earliest strain, Victoria strain is the earliest Wuhan, one of the earliest Wuhan strains, all the way down further. You can see that the numbers in the orange are decreasing, and that means that the ability of the vaccine, this is the AstraZeneca vaccine or the Covishield vaccine, the ability of the vaccine to neutralize the virus is decreasing. It still is effective, but how effective it is decreasing, and that decrease is now getting a little dramatic with BA4, BA5. So we are at the cusp of the next chapter where BA4, BA5 are likely to increase. Immune responses are likely to be more muted. The world is going to see probably more surveillance, more boosters that are specific to BA4, BA5, continued sort of alerts. And so it'll be an interesting world. But I'm thankful for all our partners, our philanthropic partners, BBMP, Karnataka government, everybody who contributed to this effort, that we were able to monitor and contribute substantially to INSACOG's efforts and supplement INSACOG's efforts. And over the last 12 months, and hopefully we can do similar sort of contributions over the near future as well. Thank you so much.
Thank you, Dr. Ramesh, for providing an overview of the COVID sequencing effort and for walking us through the evolution of the virus from July 21 to June 2022. Now, I would like to introduce our next speaker, Dr. Ashwath Narayan, Honorable Minister of Electronics, Information Technology, Biotechnology, Science and Technology, Higher Education, Skill Development, Entrepreneurship, Livelihood in the Government of Karnataka. He is a member of Karnataka Legislative Assembly from the Malleshwaram constituency. Dr. Ashwath Narayan received his MBBS degree from the Kasturba Medical College, Mangalore in 1995 and joined politics in 2004. Dr. Ashwath Narayan also served as the 8th Deputy Chief Minister of Karnataka from 20th August 2019 to 26 July 2021. I request Dr. Ashwath Narayan to share with us his perspective on managing the pandemic. Please give a big hand for Dr. Ashwath Narayan.
Namaskara. Good evening to all the invitees, dignitaries on the dais and off the dais. Today is an occasion where we're celebrating the efforts of the people who are involved in the COVID genome sequencing and surveillance. To recognize their efforts and appreciate their efforts, how much they were able to make a difference, all of us know. Really, this commendable efforts, what has been made by the biotechnologists, microbiologists, and people from many other sectors, from medical background, and the institutions involved. Probably in this direction, on the dais we do have from the Strand Life, Mr. Dr. Ramesh, who is the CEO of the Strand Life, Ramesh Hariharan, and Dr. Vishal Rao, and Dr. Rajesh Sundareshan from Indian Institute of Science, and our Dr. Bal Subramania from BBMP, and from InCovid Support, Naga Karthik, and many other people. And we have Jagdish Patankar, who is from the main event organizer for all our events in the state of Karnataka, all the most important, from biotech to IT, BT, science and technology, and nanotechnology, all possible events. It's been organized by MM Media, the promoter of that company. He's been there for all our events. Dear friends, Dr. Ramesh Hariharan made a presentation from where the COVID variants, how it started changing month on month, or the few months or 3 months or 4 months, the variants is to keep changing, as all of us know. The behavior of each virus is to be different. Some is to act on lungs, some is to act on the throat. In different areas, the behaviors were totally different. Some were oxygen dependent. The treatment line is to be different. So it was really challenging time being in the helm of affairs. We had a big challenge. Probably understanding the challenges, we were more into firefighting than looking into the strains, what kind of sequencing, what kind of behavior. We never used to much look into these aspects. We were only, into the firefighting mode, how to manage the patients, what to give the treatment. Even for doing that, without knowing the variants, particular variants, it was really difficult. Which geography, which place, what kind of variants were there, and it is to vary from place to place. And when we had Delta, England had something else. So the place of geographic location also had different challenges. Probably the timing was very important, which month, what variant was there in which place. So we keeping in this mind, and we could do a lot of things, and we constituted the genome sequencing committee in the month of June 21. We were requested to immediately constitute a committee. After constituting this committee, they worked along with the Strand Life to accelerate the entire process. They collaborated and partnered with the Strand Life, and many other important doctors were part of this committee. They took up the job of sequencing, which was not done in the entire country. Probably only few institutes were doing it. So getting the sample from across different geography and doing the genome sequencing was very important. Probably even without expecting anything from the government. They just wanted the samples. Enable and allow them to do the genome sequencing was the most important thing. Please permit us to get the samples. We need to access those samples. So immediately I took the decision as a chairman of the state COVID subcommittee, cabinet subcommittee. I was the chairman, managing the entire COVID. So immediately we constituted the committee and told our staff, our department, to immediately provide them with the samples. Probably genome sequencing of these numbers, probably even in most developed countries, they have not done such a big numbers. That's what I was told by Vishal. He had told me, no, the numbers what we have done is very big numbers comparatively to any other country. Is it the fact, Ramesh? Yeah. And more so, they took the support from various industry, like in COVID support. They came forward, the ACT came forward, and they got many other organizations to fund this entire program. The kit, what was required for this genome sequencing, it was little bit costly, for around 5,000 rupees for each sample. Probably it was costing around 5,000 rupees. And government, we had decided to open 6 genome sequencing units across the state. Probably for the establishment only, I think probably it is completed. Their entire timeline was consumed for only for procuring, establishing. Before we could do any genome sequencing, the INSACOG and our Strand Life, BBMP, government, our COVID sequencing and surveillance committee took a lot of initiative and did a lot of things even before we could install the genome sequencing event infrastructure across the state. We had identified 6 places in 6 medical colleges we wanted to put up. Before we could start, it was taken much time. But as a responsible people, nobody had expected them to come forward. Since being in a responsibility, their learning, their ability, capabilities, their concern for the society made them to come forward, do the right things. What is needed? Having a lot of knowledge, not serving or intervening in a right time. What the learning has got meaning. There's no meaning in how much we learn if you don't intervene in the right time, don't serve the purpose. So what is the point having any big institute, higher learning, gaining so much of knowledge? Ultimately, it should serve the purpose. Understanding and being so responsible without expecting anybody to ask them, come do this. They didn't expect anybody to ask. Nobody asked them. Nobody asked the gentlemen who are on the dais. They were not asked to do this. They came forward. Understanding the gravity of the situation, what is the real challenge, what needs to be done. We need people like this, people who are very committed, responsible, and understanding the need of the hour. I request all of you to give them even all of you have been part of it. Probably I think whoever is assembled here, all of you have been part of the team, I think. By representing on your behalf, probably we have them to represent on all of your behalf. They've been leading from the front. I really appreciate the efforts being made. And further, we need to work how we need to manage, because neutralizing effects of the present vaccination, treatment, what new variants will be getting, because the B5, what we are seeing, it is accessing the lungs, again dangerous. I've seen what is the impact of this virus. Personally, I've been infected more than 4 times. More than 5 times. I came to know only 4 times, and more than that probably. We could see how badly we were affected, and we were on the field exposed to everybody. And Delta had a very bad time. I had a very bad time with Delta. Delta gave me a hell of a life, and I was badly infected. Even then, we had to face all these things. It's a part of the society. We face a lot of challenges, and we'll be in a very hostile condition also. We need to work. But anyhow, this is not going to end. Probably with what I have seen, what I heard, this will be going on for many more years. This will not stop here. But our nature of working is knee-jerk reaction. Always we tend to work with emergencies. We need to prepare and ensure nothing goes wrong. We have so much of human resource, so much of infrastructure needs to be scaled, and how to handle, how to address these challenges. The preparation has to be really good. Otherwise, no economy can be stable. Nobody can look up to us. So there's a great responsibility on us, on all of us, and more so on the government, because governments need to think ahead of the time, and we need to prepare, and we need to lead, and we need to do right things. What is the need of the hour? We need to understand. Probably many things we are trying to do, sir. Probably we need to do it rightly, working with each other, with convergence, not in silos. We need to look up to each other as a society, institutions, all the institutions work in silos, because nobody has got more so we have many a times trust deficit. We are not comfortable with each other. We don't look up to each other. We tell we are not comfortable with this politician. We are not comfortable with this organization. We are not comfortable with BBMP. We are not comfortable with government. We are not comfortable with many other. We have many professional problems, institutional problems. So many parastals, we have many challenges. But overall, how we did things now at present, we need to do things in future also. Please do understand whatever needs to be done from the government. Definitely we are serious about it, and we are willing to address all these challenges. And we want to take it head on, and we want to resolve. We want to be the best possible responsible government which is for the people. And we are willing to do whatever needful, and we'll continue in this journey, and more so from biotechnology, whatever needs to be done from the biotechnology department or our science and technology, whatever you need, definitely we'll collaborate, partner, and we'll work together to address all these challenges, and whatever we have learnings we need to put into use. I really appreciate all of your efforts. Today is a day where we need to recognize what has been done and what we can do in future. So with this strength, we need to see how best we can address the challenges, and definitely there are many more challenges. So what could be the reason, I don't know, but there are many challenges, but we need to face it and overcome all these challenges and ensure that all our people health and life is protected. Many people, innocent people, lost their lives in this process. Helplessly, they were totally helpless. Many people were helpless. They were ignorant, helpless. Many times they were not getting access. It was really very challenging time. I really feel sorry for them. And it should not happen in future. It's not somebody has to do it. All of us need to be there. All of us need to responsibly work. We need to make system work, and we need to ensure the safety, security of each and every person. Let us all work together, very responsibility, with all the commitment. It's our duty. Society looks up to us because we are having so much of knowledge, we have responsibility, we have position, we have organizations to cater to all their needs. And I thank once again all of you. Without taking much time, I can assure you and reassure you, we'll be with all of you all the time. Whatever support you want, whatever might be the support, let us know. Our Honorable CM is a very learned person and is a very, responding person. Definitely our government will respond to all of our suggestions and will ensure a right system is in place to address all these challenges. And there are many emerging challenges also. Yes, the virus keep mutating. We keep getting many variants, and it can behave however it wants, and you can get new ones, old ones, variants, whatever might be. Let us all be prepared. And Bangalore is a place where the entire country is looking up to us. We need to work in this direction. I thank all of you for participating. Probably we should add bigger numbers. Only people who are involved in these efforts only are here. We should have had more people who should have come forward and appreciated the efforts, whatever all of you have done. Probably this was not the right place to conduct. And I feel the academic institutions or many other places would have been the right place, because we want people to know how things are happening. See, we are not far away from many challenges. Already we are in a time bomb. Already we are in a bomb. How it is going to explode, what is going to happen, God only knows. It should be issues where all the people need to understand. This is more serious than any of the things happening in the society. Any other events or any other programs, much more important than any other events or issues going on. This requires a topmost importance. This is the number one priority. Entire society, how ignorant, how careless we become, just see, sir. Little bit safe we become, we don't care for all these things. Still people don't know where they are, what kind of challenges are we are going to foresee. People don't understand. Probably we need to sensitize, we need to talk very strongly, not for ourselves, for our people, for our society. We need to strongly convey and communicate very strongly. Let us do it. I will be with you. Let me know in future if you want to organize any program, let us know. We can support in much more better way. We can organize in a much more bigger way. People celebrate, people should really celebrate, really recognize and appreciate all of your efforts, what you have done. You are the real heroes. The real heroes are you people. I thank all of you. I wholeheartedly thank all of you, and we'll continue in this journey and do our best. Namaskara. Jai Hind, Jai Karnataka.
ಅದ್ಭುತವಾದ ಭಾಷಣಕ್ಕಾಗಿ ಧನ್ಯವಾದಗಳು ಸರ್. ನಮ್ಮ ಮುಂದಿನ ಭಾಷಣಕಾರರು ಮುಖ್ಯ ಆರೋಗ್ಯ ಅಧಿಕಾರಿ BBMP ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್. ಈಗ ಕೋವಿಡ್ ಪೂರ್ವ ಸಿದ್ಧತೆ ಬಗ್ಗೆ ನಮಗೆ ಒಳನೋಟವುಳ್ಳ ನೀಡಲು ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್ ಅವರನ್ನು ವೇದಿಕೆ ಬರಲು ಬಯಸುತ್ತೇನೆ. ದಯಮಾಡಿ ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್ ಅವರಿಗೆ ಕೈ ಜೋಡಿಸಿ. ನಮ್ಮ ಮುಂದಿನ ಭಾಷಣಕಾರರು ಮುಖ್ಯ ಆರೋಗ್ಯ ಅಧಿಕಾರಿ ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್. ಈಗ ಕೋವಿಡ್ ಪೂರ್ವ ಸಿದ್ಧತೆ ಬಗ್ಗೆ ನಮಗೆ ಒಳನೋಟವನ್ನು ನೀಡಲು ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್ ಅವರನ್ನು ವೇದಿಕೆ ಬರಲು ಬಯಸುತ್ತೇನೆ. ದಯಮಾಡಿ ಡಾಕ್ಟರ್ ಬಾಲಸುಂದರ್ ಅವರಿಗೆ ಕೈ ಜೋಡಿಸಿ.
Thank you. Good evening to everybody, and good evening to all the guests who are here. I became Chief Health Officer in the month of November when the third wave was just starting. So I was the Zonal Health Officer during the first and second waves, and we do know that the people who are here, Ramesh sir and all, Yuvi Rao sir and all, they were doing this sequencing and they were letting the details shared with BBMP as well as the state of Karnataka. So we used to be very careful because, as you all know, the second wave was very bad when compared to all the three waves, because that was the phase when the Delta took over the earlier things, and there was sudden panicking, and as you all know, we lost many lives also. So, but for the third wave, we were very careful. We were following up with the INSACOCO recognized labs, the InStem lab, the Strand Lifeline. So these were the people who used to give us the early information regarding the changes, the mutations. So we used to plan with the clinical wing, that is the Bangalore Medical College and other medical colleges, in making policy, taking decisions like what we are supposed to do in the coming days. So when this Omicron, which was it was earlier detected in November in South Africa, and we were all worried, but INSACOCO recognized these Strand people. They were the first people to give us the information about this Omicron. I think I still remember the date, November 23rd. We got that night I was really shocked because that was the first case for the India. So we got from them, and that was the Omicron. We were worried what would happen because we had seen the second wave, which was really a nightmare. So, but because they had given the information, we were all prepared based on the first and second waves, and we had the past experience in the South Africa for the past 1.5 months. So it was a bit difficult for us because the cases, the numbers rose up very fast, because up to around 58,000, I think, the maximum in a day. But we were prepared. We had put in all things in place, like in terms of human resource, logistics, everything. We had planned in December only. We could plan because these were the people who were doing behind the screen the job which was most essential, and we could really be in a stage of well prepared kind of thing. And yes, we could, as a chief health officer, I'm proud to say that BBMP really worked hard. And third wave was, it just came and went. People did not, and not only that the third wave was so, I mean, it just came. We had prepared so much that everyday number of cases were very high, but we were in such a position that we were handling everything. The last person in that case list, we used to follow him up through teletriaging, or at least most of them were physically triaged. But if when the numbers crossed 30,000, it was difficult for us to go to everybody's houses, but still we could go contact them through telephone, and we were in a position to know the condition of the patient. So it all happened with these people. So this is genomic sequencing kind of thing. These are all the newer things which help a government body, administration to take some decisions. So I thank everybody who have worked here during the pandemic times. I really try to appreciate everybody's efforts behind this. So I personally thank Dr. U.V. Rao because I know him since many years. He had, yes, Minister Sir was saying, it is not that we called them, they came forward. They came forward with their own money. Government did not spend anything, but that helped the government. So we should support such efforts and such agencies who voluntarily come forward to help on the larger interest of the public. So I thank everybody, and I think I will not take much time, sir. Thank you all.
Thank you, Dr. Balasundar, for your insightful perspective. Our next speaker is Dr. Vishal U.S. Rao, Country Director of Head and Neck Surgical Oncology and Robotic Surgery, and the Dean for Center of Academic Research at the SCG Cancer Hospital. Dr. Vishal is well known as the inventor of the arm voice prosthesis, a low-cost speaking device for throat cancer patient, an innovation that has been considered among the 100 global social innovation across the globe and received wide recognition from BBC, Harvard Business Review, and Forbes. He is also a member of the consultative group to the Principal Scientific Advisor, Government of Karnataka, and a member of Vision Group of Biotechnology, Government of Karnataka. Dr. Vishal is also a member of Genomic Surveillance Committee, Government of Karnataka, and has been instrumental in bringing the various stakeholders together to enable effective genomic surveillance. I would now like to invite Dr. Vishal Rao to the podium and say a few words on his initiatives to bring the various stakeholders together. Please give a round of applause for Dr. Vishal Rao.
Thank you. Thank you very much. Again, without taking much time, I would like to take this opportunity to congratulate Strand Life Sciences and the entire team. The entire team of BBMP, Bala sir is here, Bhaskar is here, and the entire BBMP backend team for the efforts put during this pandemic. The state government of Karnataka, the Ministry of Health, the Ministry of ITBT, who all came together to bring this entire ecosystem together. On this day, I must start by telling you a very old quote which I remembered, which was said by Winston Churchill about Russia. He said something very interesting. He said, when Winston Churchill was asked about Russia, he said, it's a riddle wrapped in a mystery within an enigma, but perhaps there is a key. And I think that was COVID for all of us, that it was a riddle wrapped in a mystery within an enigma, and we still don't have the key. The virus is dictating the term. Every cell, it must be mutating about 300 times, and we still really don't know how it's going to plan the next couple of steps. But for me, what was interesting was that this virus weighs about 0.85 attograms. So a few grams of this virus stopped the world. Just a few grams of this virus. And that is what we saw through this entire pandemic. And on a lighter note, I keep thinking that the first symptom of COVID was not loss of sense of smell. It was loss of common sense. We all lost it. In every stage, we lost it in various junctures. And a lot of relearning. If I now look at the wave one, I really can't imagine what I did, So, I mean, I don't know what it drove us to. It drove us to a new extreme of I would say, caution. And when we became under-cautious, it made us over-cautious. So, every statement that you would utter, the next day would come wrong. I remember we were on a TV debate once. And one of the very senior scientists told a very beautiful thing. The anchor said, but yesterday you told us another statement, and today you are telling us something completely different. He said, that is science. Yesterday that was science, today this is science. He said, that was the way in which proof was changing. And we all know, on conventionally, bench to bedside takes about 17 years. And we did this in 1 year, this vaccine. And some of the joke that was carrying around was that chimpanzees are waiting for human trials to get completed so that even they get a chance. That was the pace at which science evolved, the pace at which innovations happened. And I think it is historic for us to witness, to see science evolved at this pace. When the first drug controller signs the vaccines, preclinical studies, it would be the third or fourth drug controller who would be signing the final vaccine. But it all happened within a year. And this was advances of science, technology, talent coming together with political commitment. And one such gentleman was Dr. Ashwath Narayan, and I wanted to genuinely say this, that a true pandemic CEO is a person who stands from behind, not a person who's in front. He was a person who stood behind, and I remember I was recollecting to him that day when we all went to tell him that the Genomic Surveillance Committee was to be committed. Dr. Chandru was there, I was there, a few experts from Indian Institute of Science were there, many of the people from the state government, health ministry were there. I fondly recollect Dr. Ravi sir, our chairman of the Genomic Surveillance Committee. And he asked all of us, why are you all here? And we said, I think it's time that we put together the State Genomic Surveillance Committee, because INSACOG had a lot of burden on it. And this is an entire country. So we said, since health being a state subject, we should take some state leadership. And he said, so what are you all waiting for? And we said, we should have a collated idea. And he said, no, it is you all who have to give us direction, not we give you directions. He said, get started today and tell us what are the things you need, and we'll bring all of this together. The most beautiful thing for me to learn was, it was about collaboration, not competition. And that is the most beauty thing. But he reiterated again, all of us beyond differences got together and said, let's do this. Beyond all our differences, beyond all our minor issues and other things, we said, let's put together this thing and make a small difference to show. And today I stand very proud, and thanks Ramesh and the entire Strand team, and I acknowledge all the other partners who played this important role in genomics, NCBS, Indian Institute of Science, and I think IGBT was there, and many other organizations. I'm sorry if I'm missing a few of them. I think the role that was played of collective approach, of beyond competition, collaborating, was the biggest lesson that we learned through this pandemic on how we could make a difference. And I was telling Honorable Minister that we, during this period, were sharing some learnings from us to the NVAP committee in UK, the New Variant Assessment Platform Committee, We were having interactions with CDC Sphere. I was talking to the global director, Nathaniel Smith, and I was telling him about our model. And they were excited. They were excited to say, oh wow, we all could do it. And we also wrote a white paper during that period to tell how India could actually inspire other countries who think that this can't be done. Because there were 140 countries at least, I would say confidently, who said this can't be done. Genomic surveillance is not our kind of, and we as a state were able to show through this simple example of collaboration what many countries simply couldn't do because they did not have the right, I would say, spirit to fight this and say that we should do this. And the idea was actually learning from, as Ramesh rightly showed in the slides, you see December 2019, it was the Wuhan strain. December 20, it was the Delta strain. December 21, it was the Omicron strain. I'm waiting for this December. Every year, like a new iPhone version, it is launching another one, and we are watching to see what is going to happen. The only learning for us through this genomic surveillance was, how could we be more proactive than reactive? How do we actually plan our strategies of clinical things, which we have learned now, and I'm sure if Ramesh showed the CoviServe platform, it was so beautifully done that the policymakers could on a live monitoring see how the progress of the entire virus sequencing was going on, so that you could visually through simple data visualization pick up which variant is going to change when. I think we've reached that point in science today where I think we need to now show the larger part of the world, and I still feel we are still incomplete. There's a long way to go to show the world, and these reports need to go to much more bigger corners of the world to actually tell what we have done. I was telling Jagdish sir that in the 25th Biotech Summit that's going to happen in November, I think one of the things we should tell Honorable Prime Minister was, how did we set up this? And rightfully we both were discussing that Dr. Ashwath Narayan has confirmed and said that Karnataka should house the first center for pandemic preparedness for the country over here. And I think he has already made a commitment to it, saying that this is what we will have to do, because what this pandemic taught us was, this is not enough to be reactive. He said, what is it that we can do to be prepared for the next pandemic? So wastewater surveillances, looking at active monitoring, clinical coordination, metadata, epidemiologists, data scientists, bioinformatics. I think this was a game much beyond health. Health comes much later to manage disease. And as a health expert, I'm with firm conviction telling you this. I think the foundation stone was laid by biotechnology, science and technology, and health partner to understand and interpret it. It's a game much bigger than what just one health department or medical education can own alone. And I think that's where I thank all the scientists gathered here to appreciate from the clinical side the efforts you all have put in to actually bring science and technology to this forum. And hopefully in the coming years, India will see more innovations, more research in this area to put the flag for creating India in a much bigger way and making India in a bigger way. Thank you very much, sir.
Thank you, Dr. Vishal Rao, for giving us an insightful speech. Our next speaker is Dr. Rajesh Sundaresan, Professor in the Electronics and Communication Engineering Department and the Dean of the Division of Electrical Engineering, Electronics Engineering and Computer Sciences at the Indian Institute of Science. Dr. Rajesh did his PhD and MA in Electrical Engineering from Princeton University and his BTech from IIT Madras. Dr. Rajesh research spans the area of decision theory, communication, computation, and control over networks, cyber-social systems, and more recently, data-driven decision framework for public health responses. Dr. Rajesh has worked extensively on predicting the spread of the virus in Karnataka using modeling approaches coupled with sero-survey data. I request Dr. Rajesh to share with us his experience in modeling the trajectory of the virus in the Karnataka. Please give a big hand for Dr. Rajesh Sundaresan.
Thank you for the invitation to speak here. Can we have the slides made visible? Thank you. Let me just see if it goes forward. Yeah, the pointer is not working. Yeah, okay. So I'll speak about the COVID-19 serology surveillance efforts. There will be less of modeling and more of information about the serosurveillance than the modeling effort. So this particular effort was a multi-institutional collaborative work, and I'm just a representative that is up here to speak about the work on behalf of all the people that have put together these serosurveys. Okay, for the general audience, here is a very quick overview of what serology surveillance is. It's a test that looks for antibodies in blood. So in some sense, it's a little more classical. We are not dealing with genomic surveillance here, but just surveillance based on blood. And we look for proteins that help fight the infection, that's the antibodies. Now, if you find these proteins, then the person has had infection in the past. What these serology surveillance efforts give is they provide a more complete picture of the number of infections that have occurred. And they give this across time, across space, and also across population strata. And if we get this more complete information, it helps us understand, for example, also the risk factors for disease from the data that we have across population strata. Also, it helps us guide control measures. And I'll give you one example which stemmed from the serosurvey modeling and then some concrete measures. What is this more complete picture? When you look at case trajectories, the case trajectories we all know are biased. They are biased because they are usually keyed off index cases and then contact tracing starting from these index cases. Consequently, we have far fewer number of cases identified, whereas the infection level in the population may be higher. And the infection level in the population is the one that is important to understand in the event an outbreak occurs, to what extent does the population have immunity and so on. This gap between cases and infection is measured in the form of a ratio. It's called the cases to infections ratio. More appropriately, infections to cases ratio. For every case, how many infections were there in the population? So that's the cases to infections ratio. And just as there is this gap between cases and the infection level in the population, there is also a gap between the fatality rates that are measured. There is the case fatality rate and then the infection fatality rate. Because the cases are fewer in number, the case fatality rate is typically larger, and the infection fatality rate is a fraction of the case fatality rate. These are some numbers that will come up in the later slides. Now, I want to, the serosurveillance effort involved, it was a truly multi-institutional effort with about 18 institutions here and the Technical Advisory Committee for COVID-19 for Karnataka behind this particular serosurveillance. I would like to thank all of these for their efforts. This involves the work of over 700, 800 people involved in the survey and subsequent analysis. The survey that Karnataka conducted was somewhat unique. We used a sentinel survey approach. No other state had launched such an approach at that time. Also, it was a statewide survey. It was the first statewide survey in India. So you can see on that picture on the right, several dots. Those are all the locations where the survey took place. There were 290 centers. These were district hospitals, primary health centers, community health centers, and so on. In all of these places, we engaged individuals there to be able to get samples. All the districts of Karnataka were covered, and Bangalore, because of its huge population, was subdivided into smaller units. And we leveraged the state's AIDS prevention unit as well as the tuberculosis surveillance unit to be able to recruit individuals and also collect the samples. So thanks go out to them. They actually had to step away from their regular activity to do this and then go back to their regular activity. The thing about the sentinel survey approach is something that that makes it easy to use in other places. It's very easily replicable. And we did it twice in Karnataka, and the third time is actually going on right now. to conduct a survey amongst children. Now, if you compare the survey that Karnataka did with the other states, other neighboring states, I've just listed a few here. Karnataka was the first to do the statewide survey, which was sentinel-based, and this was in September. And prior to the planning for this had started sometime in April 2020. The first survey happened in September 2020. And there were 25 revisions of the plan to conduct the survey before the survey actually took place. So it was something that took a lot of time to come to fruition, but that's also because a lot of institutions were involved. So it was the first to do the statewide sentinel-based survey in India, and the sentinel-based survey, as I also mentioned, is very easily replicable. Other states have done several rounds. Kerala, for example, has done 3 rounds, covered all districts in the last one in September. Tamil Nadu has done 4 rounds. The last one involved all the districts in December 2021. Andhra's estimates were typically based on a subset of the districts. They were more focused on what is the overall infection in the state. And the last one was, in fact, based on the national survey, July 2021. Telangana covered all the districts in their January 2022 survey. Maharashtra conducted in 6 districts during the July 2021 survey. And at the all India level, 70 districts were sampled and 4 rounds were conducted. The last one was in July 2021. So in some sense, Karnataka has been at the forefront of conducting such statewide surveys. Now, the survey that was conducted in September, October of 2020, the first survey, was something that was riddled by a certain issue, which was that the cases were also high. So we are looking for past infection when we look at antibodies, but there was also active infection. We were sampling near the peak. That rectangle there indicates the period when we were sampling. And so we had to look for active infection as well. And that's something that's novel in the Karnataka sero survey. And it's something that's that must be done whenever we are sampling at the time when infection is also raging. The sampling also involved several strata. I've listed a few out here, which sort of gives fine-grained information about the infection levels at various population strata. And the other interesting thing about this sampling was that it was from a population not suspected to have COVID-19. And so it gives us how much of this virus is circulating in the population. So that's something about the Karnataka sero survey. I'll just give some insights now. At the end of the first sero survey, this was in September 2020, the kind of estimates that we had and the uncertainty windows are given there. The thing that strikes one when one looks at this is that there was significant variation across the districts. And this was very useful information for the state's health department, because there are surges that could be anticipated in the districts which are there at the bottom where the seroprevalence was low. And there could be learnings that could be gotten from areas which had already seen that infection, BBMP, Udupi and other such places. So significant amount of variation. And when we did the second survey, we were actually dealing with a situation where there were some districts where the sero survey results for the second round was higher, and some districts where the second round was lower. And this was a sort of puzzle that we had to understand before we could put out the results. Is this because of a poor kit that we had? Is this because of antibody waning, or is it because of a combination of the two? And this led to an additional study where we had to recall certain round 1 positives, bring them back. About 16% of the individuals that were called, they were positive in the first round, they were recalled, and they were retested. And when we retested, only about 40% of them were still above the detection threshold, which was a definite indication that there is antibody waning. And that is something that had to be taken into account when one came up with the estimates. And then these were the estimates by about 18th of February before the Delta wave struck. So we were roughly at around 1/3 of the population having seen COVID-19. And then the cases to infection, we were missing quite a few cases. Between 12 and 27 was the range of the cases to infections ratio across the various districts. And also the infection fatality rate, which is a fraction of the case fatality rate, ranged between 0.25% and 50%, which was a significant number. So this indicated that there is still a significant cause of worry, and then the Delta wave hit us subsequently. This is the sort of picture that epidemiologists like to look at, because it gives a lot of insight as to how the various districts are doing. On the x-axis, we see infection fatality rate, and on the y-axis, we see the cases to infections ratio, more appropriately, infections to cases ratio. Higher one is higher a district is on the y-axis, it means that more cases are being missed. Lower cases are being identified a little more diligently. On the other hand, if we are towards the right, that means that the infection fatality rate, there are more deaths per infection. Now, of course, when we see something like this, being lower on this y-axis is a good thing. So all of these have been identifying cases, but there is also the possibility that perhaps what is Dharwad, what is the reason for Dharwad's high infection fatality rate? What is the correct infection fatality rate? Now, given the large number of districts with good facilities sort of clustered in this area, it suggests that perhaps there might be migration to hospitals in these areas, and that was certainly the case that we deduced after talking to the health officials at Dharwad. So it's sort of a center where a lot of people from the rural areas come for medical treatment, and the infection fatality rate, therefore, at Dharwad is high. And therefore, at first glance, it appears that the infection fatality rate being high is something that is negative. But on the other hand, if one thinks about it a little, it actually means that the processes in that particular, in those districts are good. And this, if the districts there have lower cases to infections ratio and sort of reasonable infection fatality ratio, we also believe that there is not enough basis for the kind of spatial variation that we are seeing, that we saw in the infection fatality rate, which as a corollary suggests that the districts that are on the top left are perhaps places where cases were missed. So these things were flagged to the Department of Health and Family Welfare Services, and example reports like these were shared with the district officials. For example, this is a district summary report for Chitradurga at the end of the first survey, which said that the IgG prevalence is lower than where Karnataka is, but then the cases to infections ratio and the infection fatality rate are too high and too low respectively, which is possibly something that triggers perhaps a testing policy review. So those were the kinds of observations and recommendations that were made to the district. So every district, every unit of BBMP also got reports of this sort on which the health commissioners could, district health officers could take some action. What good is this serosurvey data beyond such comparisons and beyond such triggered actions, maybe doing a testing review, looking at perhaps the treatment protocols revision and so on? There is actually another value which is for modeling, and we made use of this, the serosurveillance data, to understand how to allocate the scarce vaccine resources that were available when the Delta was raging. The kind of things that, insights that we could get from the modeling, which was a significant effort in itself, a fairly detailed effort, but the outcome of that modeling, which also used the serosurvey data, was is it better to, the question that one could ask is it better to allocate vaccines to those places which, districts where the surge has not yet happened, or is it better to allocate it to districts surrounding places where the infection is raging, so that we have a sort of ring fencing of the infection? What this modeling study suggested is that the best way to handle it at the Karnataka level is to actually allocate vaccines in proportion to the population of that district, which means that if you look at the percentage of the population receiving doses, all the districts should all be clustered together. But that is not what we saw. This was taken sometime around September. That's not what we saw. So this was communicated to the health department once again, and also the vaccine task force, vaccine distribution task force. And the issues here were identified as that's what we want to do, allocate vaccines in proportion to the population, but there are some districts where it takes time to absorb these vaccines and ensure that there is effective distribution, effective administration of these vaccines. And so this next picture here, where all of these should go together clustered, sort of indicates how the dose 1% and dose 2% moved with time across all the districts. Ideally, we would have liked them all to be clustered together. And hopefully, next time such a thing happens, we are better prepared to handle the pandemic, and we'll be able to do things of the sort that this one suggested we should do. So this picture here is the overall picture, which is the final image of the video that I just showed. So what we did well with the serosurvey, just to come to the conclusion, is that I think we had good geographical spread. We leveraged the sentinel sites. So we got a good picture of where Karnataka was, which district was doing well, which district needed a little bit of attention, and so on. We also leveraged the sentinel sites. We did not use any new infrastructure, and we did it twice, which means that perhaps this could be a model that could be replicated by other states. The designs that we had were somewhat complex in the first round, but we corrected it in the second and ensured that we could get better analysis done on the data that we got. There are many improvements that are possible, and we should certainly take these into account. Standardization of kits for such serosurveys. We had an optimal design in terms of the tests that were administered, but not in terms of the strata, the population strata. So that's something that we should look into. We lost a lot of data because of manual transcription errors. Perhaps better automation will help us. Better automation with barcoding can lessen loss. And also the fact that the infection fatality rate across districts is so spread out is suggestive of possible improvement in auditing of the deaths, which will give us a better infection fatality rate estimate. Okay, so here are some concluding remarks. I already mentioned standardization of the surveys and replication of this across the nation. There was also clinical surveillance that was spoken about, and this is something that the Health Commissioner had indicated to all the hospitals. This actually came out of a discussion that we had with Dr. Angelique Kodzi from South Africa, who was one of the first persons to spot the Omicron there. She noticed that the cases that were coming had a pronounced change in the clinical symptoms that they were presenting. The infection was restricted to the upper respiratory tract. And that sort of led them to say that the virus, perhaps there is a new mutant, and the sequencing then led to the identification of the Omicron. This was around November of 2021. And when that was related to the Technical Advisory Committee, there was further action, and we eventually had all the hospitals monitoring for possible changes in the symptoms that the patients were presenting. Also, genomic surveillance is something that we have done quite well, 2 lakh samples or so, but we were able to catch the variants reasonably quickly. But when we compare with what UK has done, and UK has lesser population than Karnataka, it suggests that there is still some distance to go. Also, we should use our stored sera from the serosurveys more effectively. This is a fantastic way by which we can assess immunity against new variants. Whenever there is a new variant that comes, we could perhaps grow those viruses in the lab and then do lab tests against these sera that we have collected to see if the sera are effective, the antibodies are effective against these new variants. That's something that we should be doing, we ought to be doing, and hopefully we'll do it in the future. And of course, many people spoke about wastewater surveillance. So I'll stop there. Thank you very much.
Thank you, Dr. Rajesh, for an insightful speech. Our next speaker is Shri Naga Karthik from In COVID Support, who will share highlights of the magnificent work done by In-COVID Support Organization in supporting a large number of efforts towards COVID and overall pandemic responses. Please give a big hand for Sri Naga Karthik.
Hi everyone, good evening. I'm representing In-COVID Support, I think more commonly known as Crypto Relief here today. Crypto Relief came about a little over a year ago, started by Sandeep, and it started with a tweet. And essentially what happened after that is like a bunch of us, I think most of whom didn't know each other, sort of just rallied around the cause. A lot of us are here to basically try and see what is it that we could do. We definitely did not intend to be as large as we are today, but I think as a group of volunteers, we've tried to do our part in what we can to help. And we're now in the process of setting up an entity and a more structured process for this. Especially with respect to this, when this conversation came up, I think Dr. Vishal was the one who got in touch with us. And I clearly remember the conversation. So I think the first conversation when they got in touch with us was, there must be something off because no other state had allowed private labs to do sequencing. And when they sort of showed us the letter of the Genome Sequencing Committee, and I think that's when we realized how also proactive the Karnataka government was being in actually setting the stage for this to happen. And it became a very exciting project for us. When the initial proposition came, I think there was a little bit of delay from our end to get things started, but I think we're glad we did. I also remember when that initial conversation happened, they made it very clear to us because we were in from the space, and when they were explaining what this project would achieve, they also said we might actually not find anything. Like we'll do all the sequencing that is possible. But you have to be prepared for the fact that we will not find anything. But I think at least we're all glad that we went through, and it's really exciting and great to see what all has been achieved in the last year. And as Crypto Relief, we are committed to continue supporting the public health ecosystem in India. And when we say public health, we're definitely looking at things like genome sequencing, environmental surveillance, wastewater surveillance. A lot of the work around surveillance from our end, I think, is happening and starting in Bangalore. And I think we want to use the human capital and the expertise that we have here to try and see how we can take it to different parts of the country. So overall, I think it's been very, very, we were glad we could play this role. I think our role has been the smallest, but I think it set up the stage for this conversation to be had on multiple platforms like this. And I think it could set the stage to do more going forward. As a fund, we are committed to see this through to a much larger extent. I think Sandeep did mention about an all-India alliance that we're looking at for surveillance. There is a virus research institute on the cards. There is a lot of work that will happen at the district level. And we stand committed for the next few years, for a good next few years, to sort of invest in this deeply, work with state government, civil society, private players, to actually see how we can be a catalyst and take this forward. So yeah, I think that's pretty much it from my end. But also, I think everyone in the team wanted to take this opportunity to also congratulate Strand for getting into the INSACOG network, and I think that's a good achievement. And I was also asked to make sure that I tell this to the Strand team. So thank you, thanks very much.
Thank you, Sri Naga Karthik, for a wonderful speech. Thanks to all our speakers for giving us an insightful overview of the multi-dimensional collaborative efforts to manage a pandemic in Bangalore and Karnataka in particular, and in India in general. We hope that such collaboration will continue to flourish in the times to come. Ladies and gentlemen, let's give a huge round of applause to all the speakers. I will now invite Dr. Vamsi, Chief Scientific Officer at Strand Life Sciences. Dr. Vamsi did his BTech at IIT Kanpur and then received a PhD in computer science from Penn State University for his work on genome assembly algorithm. Dr. Vamsi joined Strand in 2004 and leads a team of scientists and engineers at Strand building algorithm, tools, and laboratory tests for various genomic applications, both research and clinical. I invite Dr. Vamsi to provide the vote of thanks. Please give a big hand for Dr. Vamsi.
Thank you all for attending this event today. It's my honor to give the vote of thanks for this event. First of all, I'd like to start off by thanking Honorable Minister Dr. Ashwath Narayan, and especially for his emphasis on how open the government of Karnataka is to be at the forefront of such activities and collaboration with research groups. Next, I'd like to thank Dr. Ramesh Hariharan, Dr. Rajesh Sundaresan, Dr. Rakesh Mishra, and all of these are academics, academicians, scientists, and technologists who are working in different spheres of life, but all of them repurpose their activities to tackle this pandemic head-on and shed insights into how academic research can be reoriented towards tackling a crisis in real time. So thank you all for your insightful talks on that. Next, I'd like to thank Dr. Vishal Rao, who has acted as a glue in connecting academicians, genomic surveillance committees, the government of Karnataka, and the funding agencies to try to get these activities together. A lot of this would not have been possible without the heartfelt cooperation of so many people at BBMP. I would like to thank the commissioners, Dr. Trilok Chandra, Shri Tushar Girinath, for their wholehearted support right from the beginning. I'd like to thank Dr. Balasundar, the Chief Medical Officer, who gave a talk today. I'd also like to thank Dr. Bhaskar Rajkumar and Dr. Maheshwari from the BBMP COVID War Room, who helped with a lot of the arrangements for this particular event. And also Dr. Deepak Nadik from BBMP, who put us in touch with many of the RT-PCR labs distributed across 50, 60 wards all across the city. And he was instrumental in making sure we could collect samples. So I would like to thank all of the PCR labs, the technicians, the logistics people who worked tirelessly in making sure these samples were collected on time and that we could provide timely reports. Finally, I'd like to thank the funding agencies, Dr. Shri Naga Karthik and Sandeep Nailwal, for funding this effort and showing a new paradigm of public-private partnerships where multiple disparate entities can come together in the service of a larger good. And I'd like to thank the organizers for arranging this event, and the Strand team and the BBMP team and the in-COVID teams, all of you for coming together for this particular event. Thank you all and have a good evening.
Thank you, Dr. Vamsi. Ladies and gentlemen, before closing the event, in behalf of Strand Life Sciences, I would like to thank again each one of you for taking out time on a Sunday evening and make it to this event. We are really grateful, thankful, and felt privileged to be associated with so many brilliant yet humble people around. We reiterate our thanks for the collaboration and support for Government of Karnataka and BBMP and continuous general philanthropic funding from in-COVID support. Thank you so much, and we wish you and your family stay safe. Thank you so much. Before leaving, please join us for the tea and refreshment in the reception. Thank you.